Lecture 7. Colorimetry. Introduction 1. The limits of a psychophysiological nature inherent in the process of perception require a specification of color through objective type attributes that allow us to define, in a unique and quantitative way, what we perceive, in a subjective and qualitative manner, as chromatic sensations. Colorimetry is the science that deals with methods for measuring color, based on the relationships between energy interactions with materials and the physiology of human vision, defining reference standards for which the definition and exact reproduction of a certain color. We have already seen how the colors physically correspond to the result of the interaction of electromagnetic energy with the world around us, in the form of reflected, transmitted, or emitted energy, in the visible range. Read and interpreted by the human eye perceives as a unique event. In fact, each color corresponds to a radiant flow curve that represents not a single value, but infinite spectral information dependent on two external factors, such as the nature of the observed surface and the characteristics of the illuminant, and an internal one, which is the human perception. Introduction 2. Therefore, a color measurement must take these factors into account simultaneously, while the reference to only one of them will offer an incomplete and unsatisfactory description. The clearest proof of this lies in the fact that different radiant flow curves can correspond to perfectly identical impressions of color or stimuli. This phenomenon is known as metamerism, and the colors that show this feature are called metamer colors. In essence, given a radiant flow curve, the eye is not able to read all the information contained, as the wavelength varies, but perceives of it a global result, a cumulative value, from which it derives precisely the color impression, better known as stimulus value, or tristimulus if we consider the trichromatic model of perception. Introduction 3. Example of metamerism. The same objects appears to be different colors depending on the light source. Introduction 4. The curve in the following figure, for example, describes, in terms of reflectivity P, the spectral behavior of a red panel, illuminated by daylight and red through the detectors of a colorimeter. Although the curve itself includes a detailed description of the spectral trend of the surface observed over the whole visible spectrum, it appears evident that the instrument eye will only receive a global impression of red color, because a general absorption occurs in a large part of the spectrum, with greater values of reflection starting from 600 nanometers in orange and red. On the other hand, the same curve says nothing about the typology of such a red, and it is impossible to know from the spectral curve how intense or brilliant the color it represents will appear to our eyes. Introduction 5. A red-colored panel, in natural daylight conditions, type C source, shows its own radiant flux curve that the eye perceives as a resultant global effect based on the three main stimuli of sensitivity in red, green, and blue. Color definition 1. There are three conditions: color, purity, and intensity that allow to obtain the complete parameterization of the color. 1. The color corresponds to the wavelength of spectral colors, such as violet, blue, red, and so on, to which the values of tristimulus transmitted to the brain are associated. The color is the basic component of color and corresponds to the chromatic sensation. On the one hand, it assumes a psychological meaning, association of a color to a specific region of the spectrum, and on the other it becomes a very precise physical quantity, definition of the associated wavelength. In essence, if several people can agree on the impression of the color of a color, for example on the fact that the panel in question is red instead of green, it is more difficult to decide which is the exact red color observed with respect to the infinite colors contained in the spectrum. Color definition 2. 2. The purity or saturation of a color, on the other hand, indicates how much a color observed differs from its corresponding saturated, or pure or spectra, color, as it is determined by its color. In practice, the lights observed in nature are hardly monochromatic, that is, they can be associated perfectly with a single wavelength. Instead, they are mostly multichromatic, that is, they contain a different number of wavelengths, although they can be grouped in a particular neighborhood, as in the example of the panel. Within certain limits, the dominant color remains unchanged even if the spectral range considered increases. What changes is the impression of the dominant hue that becomes, in a certain sense, increasingly faded and less certain, moving away from the dominant wavelength, until, at the extreme, any hue is lost, and therefore of color, and one enters the achromatic dimension, where precisely it no longer makes sense to speak of color but of simple luminous intensity, that is, one enters the world of grays. Color definition, 3. 3. The intensity of a color, the last of the three parameters, refers to the amount of reflected light energy, so you can have the same colors, and therefore equivalent in their chromatic components of hue and saturation, which appear more or less bright, more or less dark. This also depends on the different spectral sensitivity of the human eye, so that the colors that appear first, and therefore even brighter, are those of green-yellow around 555 nanometers. The intensity essentially expresses the achromatic component of the observed color, regardless of its hue and saturation, which are, instead, the chromed components which, for example, the brown color of chocolate in all respects shows the same hue, lambda equals 580 nanometers, and saturation of an orange yellow, almost like an orange, but with a decidedly much lower intensity value. The color factor is perceptible only within certain limits. On the contrary, for very high intensity values, as you can guess, no particular color can prevail and the saturation tends to be minimal, so that it no longer makes sense to talk about color. CIE colorimetric system, 1. The first necessary step for the definition of a normalized colorimetric method was to quantify the different reactivities of the three photoreceptor systems of the human eye during the perceptive action of a given color. This required various experiments, among which we recall the most famous of Stilesberch and Speranskaya, which revealed the reactivity of a population of individuals considered to have a normal vision, through the stimulation provoked by various CC colors of the spectrum put in direct comparison with other colors CS obtained according to the Young model, for additive synthesis of three monochromatic beams, based on a theme of primary colors, red, RS, green, VS, and blue, BS, chosen. Appropriately, figure below. Scheme for the reproduction of a color and for the experimental determination of the tristimulus coefficients. Three beams of white light, coming from three projectors with adjustable intensity, are filtered with three primaries red RS, green VS and blue BS suitably chosen and illuminate the lower part of a white Lambertian screen. At the top of the screen is the CC sample color whose characteristics are to be determined. When the observer, by adjusting the intensity of the three beams of light, no longer sees the separation between the two faces of the screen, it means that the color CS reproduced by synthesis of lights at the bottom is equivalent to the color present in the upper part. In this way, the proportions of blue, green, and red primary lights present in the CC color are determined with respect to the sensitivity of the three photoreceptors of the human eye, also called SAD-P values. It is a method of direct confrontation where the judge is not a tool but the human eye. CIE colorimetric system, 4. The results of this experiment are represented in the following figure by the three functions RS, lambda, GS, lambda, BS, lambda, called tristimulus or color matching, which describe the necessary parts, computed on all the spectrum of the visible and relative to the three red, green and blue primaries used in the synthesis, to obtain the exact perception of a given color CC. These functions therefore represent the key to realize the information transfer between the external physical reality of color and the internal perceptive reality of the human eye. CIE colorimetric system, 5. It is important to underline that the tristimulus diagrams are linked to the spectral characteristics of the primaries chosen in the reference system. In the specific case, the trichromatic model was adopted and the three monochromatic beams used were chosen with wavelengths equal to 645.2 nanometers for RS, 526.3 nanometers for GS, and 444.4 nanometers for BS. If the references were changed, the results would obviously be different. Therefore, the choice of primary colors is a very important factor and should be done with the criterion that each of them is a color that cannot be reproduced through the synthesis of the other two. CIE colorimetric system, 6. These results, obtained experimentally, showed two problematic aspects due to the specific spectral sensitivity of the cones in the human retina, namely, the impossibility of obtaining the white color with equal weights of tristimulus RS, GS, BS contrary to the assumption of the starting trichromatic model. The presence of negative values of tristimulus due to the behavior of the function of the red RS, lambda, in the spectral range of the blue-green, between the wavelengths of the two primaries GS, BS, contrary to the hypothesis of starting only sums of the three monochromatic beams. To overcome these inconsistencies, the decisive steps were taken in 1931 by the CIE, Commission Internationale de l'Éclairage, through the adoption of new units of measurement and three new reference primaries. CIE Colorimetric System, 7. In order to overcome this imbalance, the units of measurement of the different red, green and blue components used in the additive synthesis were modified differently, so as to derive new functions of chromatic correspondence Rs, lambda, Gs, lambda, Bs, lambda, as represented in the following figure, where the values of tristimulus R, G, B necessary to define the sensation of white are equivalent at this point. The average tristimulus functions Rs, lambda, Gs, lambda, Bs, lambda, proposed by the CIE in 1931, resulting from the application of three new primaries R, G, and B centered on the following wavelengths: 700.0 nanometers for the red, 546.1 nanometers for green and 435.8 nanometers for blue, which ensure the equal distribution of the tristimuli in the definition of white light. CIE Colorimetric System, 8. Then the CIE adopted three new reference primaries that were closer to the triplet of the additive primary colors red, green and blue, although more distant from the spectral sensitivity peaks of the cones of the human eye, with the following wavelength characteristics: R for red at 700.0 nanometers, G for green at 546.1 nanometers and B for blue at 435.8 nanometers. The main use of the tristimulus functions Rs, lambda, Gs, lambda, Bs, lambda, is to contribute to the quantitative determination of AC, RGB, color, based on the three scalar values of tristimulus R, G, B, which include, by definition, also the influence of the other two external parameters that intervene in the process: the spectral function of the radiant flux of the illuminant and the reflectivity or transmissivity, depending on the case, of the observed surface. CIE Colorimetric System, 9. In fact, the impressions of red, green and blue defined by the values of tristimulus R, G, B are the result of the integration of the functions that describe the process on the whole spectrum, between 400 to 700 nanometers, as follows. The resulting total luminous flux, or color stimulus, will be given by, measured in, W, where Fr, Fg, Fb are constants, proportional to the unit quantities of the adopted trichromatic system which, for the observations made, are worth 1.0000, 4.5907 and 0.0601 respectively. CIE Colorimetric System, 10. The definitive tristimulus functions X, lambda, Y, lambda, Z, lambda, adopted by the CIE in 1931 as standard functions, result from the interpolation of the experimental ones, modified in order to ensure the equal distribution of the three stimuli in the definition of white color and in order to keep all the values of the functions themselves positive. Luminous and illuminant sources, 1. Considering the importance of the spectral and power characteristics of the illuminating radiant flux, the CIE, also in 1931, defined some lighting standards corresponding to different light sources, including artificial light, direct sunlight and, finally, daylight diffuse, following figure. The sources are distinguished on the basis of the so-called color temperature, corresponding to the absolute temperature, expressed in degrees Kelvin, of a black body that emits a radiant luminous flux with similar chromatic characteristics. It is recalled that in photometry there is a precise distinction between the source, understood as a body that emits radiant energy, and illuminating, understood as a spectral distribution of radiant power coming from the source. Luminous and illuminant sources, 2. Spectral characteristics of the main CIE standard illuminants, normalized at 555 nanometers. Source A. Tungsten filament at an absolute temperature of 2854 K, source D100, direct sunlight for an equivalent temperature of 4870 K, source C, average daylight, under overcast conditions, for a temperature of 6770 K. Luminous and illuminant sources, 3. The most important standard sources are defined by the characteristics of the following illuminants. Type A. It represents the light of a radiant black body at an absolute temperature of 2854 K and is equivalent to a source made with a tungsten filament. Type D100. It represents the direct light of the sun for an equivalent temperature of 4870 K and is realized by combining source A with a particular filter. Type C. It represents the average daylight, under overcast conditions, with an equivalent temperature of 6770 K. It is also realized by combining source A with a particular filter. Type W. It represents the light that can be obtained by adding together the primary components of a colorimetric system. It is often also indicated with E. Type D65. It represents the average daylight in the interval between 2 hours after sunrise and 2 hours before sunset, for a temperature of 6500 K. Its power spectral distribution is very similar to that of illuminant C and is quite stable in the visible, for a temperature between 6000 and 7000 K, corresponding to the average daily variability value. To date, it has not been possible to simulate satisfactorily a source of this type in an artificial way, so that in 1980 the CM developed different light simulators, according to the various phases of the day. CIE chromaticity diagrams and coordinates. 1. The definition of the tristimulus values, both R, V, B or X, Y, Z, allows defining triplets of values that uniquely identify a certain color with respect to the reference primaries R, V, B or X, Y, Z. In both cases, the most appropriate way to graphically represent triplets of values is that of the color triangle whose vertices are placed the three reference primaries and where each point inside the triangle represents a color defined by the weighted values of the three primary components R, G, B or X, Y, Z. CIE chromaticity diagrams and coordinates. 2. The equilateral triangle is the most appropriate representation for triplets of tristimulus values, both in the system R, G, B with real primaries that X, Y, Z with imaginary primaries, A. In order to include also the real colors with negative values of negative tristimulus, the triangle RGB is modified in a figure with an external extension with respect to the BG side, B. Finally, the relationship between the triangular diagrams of the two systems R, G, B and X, Y, Z, C. CIE chromaticity diagrams and coordinates. 3. An appropriate way to relate tristimulus values to each other is normalization with respect to their sum. In this way, we have the following expression systems. OR, where R, G, B and X, Y, Z are the chromatic coordinates, or trichromatic coefficients, and represent the normalization of each tristimulus value with respect to their sum according to the two CIE R, G, B and X, Y colorimetric systems, Z respectively. The chromaticity diagram illustrates a number of interesting properties of the CIE XYZ color space. The diagram represents all of the chromaticities visible to the average person. These are shown in color and this region is called the gamut of human vision. The gamut of all visible chromaticities on the CIE plot is the tongue-shaped or horseshoe-shaped figure shown in color. The curved edge of the gamut is called the spectral locus and corresponds to monochromatic light, each point representing a pure hue of a single wavelength, with wavelengths listed in nanometers. The straight edge on the lower part of the gamut is called the line of purples. These colors, although they are on the border of the gamut, have no counterpart in monochromatic light. Less saturated colors appear in the interior of the figure with white at the center. It is seen that all visible chromaticities correspond to non-negative values of X, Y, and Z, and therefore to non-negative values of X, Y, and Z. If one chooses any 2 points of color on the chromaticity diagram, then all the colors that lie in a straight line between the 2 points can be formed by mixing these 2 colors. It follows that the gamut of colors must be convex in shape. All colors that can be formed by mixing 3 sources are found inside the triangle formed by the source points on the chromaticity diagram, and so on for multiple sources. CIE chromaticity diagrams and coordinates. 6. An equal mixture of 2 equally bright colors will not generally lie on the midpoint of that line segment. In more general terms, a distance on the CIE XY chromaticity diagram does not correspond to the degree of difference between 2 colors. In the early 1940s, David McAdam studied the nature of visual sensitivity to color differences, and summarized his results in the concept of a McAdam ellipse. Based on the work of McAdam, the CIE 1960, CIE 1964, and CIE 1976 color spaces were developed, with the goal of achieving perceptual uniformity, have an equal distance in the color space correspond to equal differences in color. Although they were a distinct improvement over the CIE 1931 system, they were not completely free of distortion. It can be seen that, given three real sources, these sources cannot cover the gamut of human vision. Geometrically stated, there are no 3 points within the gamut that form a triangle that includes the entire gamut, or more simply, the gamut of human vision is not a triangle. Light with a flat power spectrum in terms of wavelength, equal power in every 1 nanometer interval, corresponds to the point x, y equals 1/3, 1/3.